next speaker will be Ricard Alert, and we'll talk about capillary interactions, organized bacterial colonies. And you have quite something to live up to.
It means to confuse.
All right. So I wanted to start by thanking Christine and Raul for organizing the workshop and putting this together, and all of you for being here, even last day. And so I initially thought I would talk about capillary interactions and telling you how they organize bacterial colonies, and it's still true. But I also want to talk about something else. So I'll start with the capillary interactions, but then I'll also talk about active colloidal particles and tell you about how they flock by turning away from one another. And this is essentially based on discussions with people here in the workshop, and I found that many people were interested in this, so why not talk about it? All right, so let me start with the capillary interactions first. So first I want to acknowledge who did the work. And so this is a collaboration between theory and experiments, and the experiments were done by Matt Black in the lab of Josh Shavitz in Princeton. And the theory we started together with Ned Wingren a long time ago when I started my postdoc, and then Chen Yi joined us and did all the nice work that you'll see today at the end of, more recently. All right, so what I want to tell you about is the life cycle of this bacterium, which is called Myxococcus xanthus. I will refer to it simply as Myxo from now on. And so this is a soil bacterium, and it lives always in big colonies. And so what it does to live is essentially, imagine that you get a few spores of this bacterium. These are spherical cells that are metabolically inactive, and they can survive without food for very long times. They are resistant to starvation. They have no metabolism, essentially. And so then what happens is if you give them food, they can undergo a process known as germination, whereby they transform themselves from spores into rod-shaped cells, and they become active. They turn on metabolism, and they start moving on surfaces in the soil to prey on other bacteria. And so this phase in which they swarm and prey is called swarming. And then what happens is when they run out of food, they aggregate into very dense two-dimensional colonies of bacteria, and then eventually from these very dense colonies they form three-dimensional structures that look like this, like little droplets, each of which contains hundreds of thousands of individual bacteria, and these structures are known as fruiting bodies. And what's interesting is that these bacteria turn themselves back into spores at the top of these fruiting bodies, and this allows the entire colony to survive starvation. Okay? So this is the collective life cycle of this bacterium, and what I want to tell you about today is, first of all, how do you go from swarming into these very dense two-dimensional aggregates, and then to start with, I'll just say a few words about how these two-dimensional aggregates form fruiting bodies. So I want to start by showing you how one of these two-dimensional aggregates looks like, and so this is how they look like. This is a single layer of bacteria imaged under the microscope. You see that these bacteria are rod-shaped, and so they are packed at these very high densities, and as a consequence of that, they align with one another. And so essentially what you're seeing is a phase of matter with orientational order, and that's a liquid crystal. It's in particular a nematic liquid crystal made of bacteria. And so what we found previously by studying this system is that there are also, of course, topological defects associated with the nematic order, and it's precisely at these topological defects that these bacteria form new cell layers that allow them to form fruiting bodies. Okay? So that's what we did in the past. What I want to focus on today is how do these bacteria form such nematic layers in the first place. And so we started getting interested in this by looking at the edge of one of these holes that you see in the colony. And so if you look closely at the edge of one of these holes, hopefully you can see that there's a halo that surrounds it. Okay? There's this kind of halo around the edge of the colony. And what I want to try to show you today is that this halo is actually water. That this is a meniscus of water, because there's water covering the entire cell colony, and you see it here at the edge. And so to convince us of that, we kind of got convinced of this when we saw events like this. So you see here two groups of cells, and they'll move around. You see their halos around them, and eventually these two halos, which are menisci, will come together, and then you see this very strong capillary attraction between the two groups of cells. Similarly, if you now look at another example of now a single group of cells, they try to split, but then eventually, boom, they get pulled back by what we think are capillary forces, and eventually on a second attempt, they manage to split. Okay, so by looking at events like this, we started thinking this looks like capillary forces, this must be menisci of water. And so then we started studying this, and the way we thought about it in a model is what follows. So what we did was to develop a hybrid model in which we describe the cells as spherocylinders. So this is the discrete aspect of the model. And then we describe the surface of this meniscus as a continuum field. So that's a continuum aspect of the model. And it's this height field of the water surface. And so then what we do is we write down the free energy for this height field. And we say the energy comes from surface tension. And I pay proportionally to the surface area of this interface. And then there's another cost, which is the cost of extracting water from the substrate. Because essentially these bacteria live on a hydrogel. And so the hydrogel is very hydrophilic. Extracting water from it is costly. And this comes at the cost of this osmotic pressure P. And so this is the cost of extracting water and forming these menisci that are above the substrate. Okay? So then from this, what we do is we run simulations in which we minimize this free energy under the constraint that it cannot go, that the surface cannot go below the cells. And in this case, we get, images like that. And so essentially what this tells us is that this decay of the water interface of the meniscus is given by the capillary length, which is defined in terms of the two parameters of this theory, which are the surface tension and this osmotic pressure. Okay? So this gives us the decay length of the meniscus. And so then the nice thing is that we can test this against experiments because they have a very interesting microscope that is called a profilometer that can measure height profiles very accurately. And so this is an example of this measurement. So this is a single cell. You see its profile and you see the halo around it, which we think is water. And so then we can take a profile along a direction perpendicular to the body axis. And so if we do that, the profile looks like this, and the continuous line is essentially a fit of the theory that I told you about. And so from here, we can fit the capillary length, which is this ratio between surface tension and osmotic pressure. Okay? And so at this point, we said, okay, this really looks like it's water, but we want to test it, so we need to be able to modify some of these parameters and see if the theory still fits. And so for a very long time, I was insisting that we should try to modify surface tension, and the experimentalists tried very hard, but it turns out every time you modify surface tension, you have to use a surfactant, and in all cases, cell died. Okay, so this experiment was a massive failure, and this is what happens sometimes when a theory is proposing an experiment. Until, the project was stuck for a long time, until Josh Shavitz actually realized, well, if modifying surface tension is such a pain, why don't we modify the osmotic pressure? And that's what they did. They came up with a way of varying the capillary length by modifying osmotic pressure through a device, which is very simple. It's just you take your sample with the bacteria on top, and you just attach it to a water reservoir, and you can tune the height of this water reservoir, therefore changing pressure. Okay, this makes water more or less available to the cells to extract from the surface. And so doing that, you essentially have a pressure which is some initial pressure and then changed by the hydrostatic pressure of the water reservoir. And so this is how the experiment looks like. As you can see, very fancy. There's the microscope here with the sample in it, and then you just have this pipe that connects the sample to this water reservoir. You can have several of them at different heights. You can even dynamically change the height of the water reservoir during the experiment. Question?
So the substrate is a porous agar?
It's porous agar, exactly.
Wetting, ideally wetting.
Exactly, yes. So then with this experiment, what we could do is to essentially have different heights of the water reservoir and therefore generate different pressures. We know the surface tension of water, so we could pre-calculate what we thought would be the capillary length. This is pre-calculated. And then we can also obtain it from the fitting to the meniscus that I showed you before. And this matches, okay? So this, I think, actually shows that what we see is consistent with the model we had and with the presence of water in this menisci. And we did this for different types of bacteria, and of course, they all fit, okay? This is just a physical effect that is independent of the bacterium that you use. All right. So then the interesting thing to do next is to try to understand capillary interactions. And so for this, what we did was to add dynamics and write down how the cells would move. And so what we have is an equation of motion for the cells, which experience some friction with the substrate. Importantly, this friction coefficient here is not just a scalar quantity, but it's a matrix because the cells are rod-shaped, and so they have a different friction along their body axis than perpendicular to it. There's anisotropic friction. And then we have steric interaction forces between the bacteria, and then capillary forces that are computed in this way, obtained from the tendency to minimize this free energy of the water-air interface. Okay? So these are capillary forces in a model of this rod-shaped object. And then we also have torques that tell us how the orientation of these rods changes, again, because of steric torques of interaction between bacteria, and because of capillary torques that, again, follow from some expression derived from this free energy. Okay? So now we can study capillary interactions between bacteria, and so that's what we did next. Here's an example in which we have non-motile bacteria, so these bacteria are completely non-moving, not self-propelling, and they just happen to be close together, and eventually fluctuations make the two menisci come together, and the bacteria attract each other. Okay? And so we can track the angle, the relative angle between these two groups of cells and their distance as a function of time. And you can see that the angle increases, consistent with what you hopefully saw, which is that the cells first turn towards the other ones, and then they become straight. And so the angle first increases and then goes down to 0 once the cells become parallel. And the distance just monotonically decreases. All right? And we can fit this with the model that we had. And the fitting parameters here is we know everything but the friction coefficients. And so we fit the parallel and perpendicular friction coefficients through these curves.
How many Myxo are there in each of the clumps?
Here it's 2 and 1. Maybe I can replay it.
Oh,
so you see
exactly
there's
one Myxus. Oh yeah.
this
okay,
is just
got it.
1 and this is 2
Now I see.
actually. Yeah. So we couldn't get an example of 1 and 1, so we got 1 and 2. So hopefully now by seeing it again, you saw this first increase in angle and then decrease down to 0. All right, so now the next thing to do is to add self-propulsion, right? Make it active. So we have a model of rods interacting via capillary forces, but now we want to make the rods self-propelled. And so we just add a term that does that. This is a self-propulsion force along the direction of the body axis of this bacterium, Ni, and then this is a stochastic variable that takes into account the fact that these bacteria reverse their direction of motion every so often stochastically. And so there are stochastic reversals that we take into account through this stochastic variable here. And with this, we can then simulate what happens at different regimes. So first I want to show you,
The chi is just sort of telegraph, I mean, it's 1 minus 1.
Exactly, dichotomous noise.
Poisson or something.
Poisson dichotomous noise, yes, exactly. So I want to show you what happens in simulations of this model, first at extreme regimes. So if the capillary forces are very weak, then what you see here is a collection of cells that are moving in this yellow background, which is the height field, which is very high everywhere, because the meniscus is very wide. It's very wide, but very weak, okay? And so this meniscus generates no attraction almost. It just allows cells to move along. And so the cells form a gas that acquires nematic order over time, but it's still a gas. Now, instead, if we go to a regime with very strong capillary attractions, then the cells attract very strongly. The menisci are very small. Now they only reach this, top of the cells, which you see in yellow. And then the self-propulsion forces are unable to deform the water interface, and therefore the cells just clump together in droplets, and they cannot move much. Okay, so that's the other extreme regime. And what's interesting is what happens in between. In intermediate regimes in which the capillary and the self-propulsion forces are similar in magnitude, then the capillary forces still make cells come together, but the cells can deform the interface, and then they can elongate along this axis of nematic order and form streams, nematic streams, which are actually very close to what's seen in experiments. Question?
So the nematic order in the gas is kind of silly, right? I mean, you
Exactly.
twice. So you just meet someone and then align with them.
Correct, yeah. That's just through steric interactions, yeah.
Yeah, but also you don't decorrelate ever. If you align, you remain alive forever, right?
Exactly. Well, you can also add rotational diffusivity, and then you decorrelate a bit.
Yeah, so you would say simulations of mix without spatial diffusion often look very similar to.
Agreed, yeah. All right, and then one more thing that we can do is staying in this same intermediate strength of the capillary forces. What we can do is play with the self-propulsion, with the reversal frequency, and tune it down. And if we have a very low reversal rate, then what we see is little flocks, so just little polar clusters of cells that are very persistent and just keep moving. They can meet another cluster, but because the self-propulsion forces are strong enough, they can overcome it, and they can overcome the capillary forces and just move past it. And so this is also what we roughly see in experiments when we have mutants that do not reverse as often. All right. Then what we could do next is to try to, question.
Can you say in a different tool how you actually simulate this? Because with both the motility and the meniscus.
Sorry, can you say again?
Can you just tell us a little bit how you actually simulate this?
Yes. So at each, so essentially we lay down the cells. Then we run this constraint minimization algorithm to determine what's the water surface, which we relax to equilibrium at every time step. And then from that, we calculate capillary forces and update the cell positions and do it again. So what we did next was to try to test these ideas in experiments. And so what they can do is dynamically change the availability of water. And so here's an experiment in which they start with the water reservoir up, which makes water available. And so in this case, cells can freely move within this very wide menisci that produce very small capillary forces. But then at some point, we can lower the water reservoir, and then suddenly what happens is this. And so you see dynamically capillary forces increase, and cells condense into these dense aggregates that then develop nematic order, and as a result elongate, and you get this set of nematic streams. All right. And so the other thing I want to show you is that when you get these nematic streams, then of course the nematic order increases, and this we also see dynamically in one of these experiments in which we change the water reservoir. So we start, let's say, with wide meniscus, with relatively short meniscus, and then we have a relatively high nematic order. Then we flood the system with water, and so we lose nematic order, and the system becomes this roughly isotropic gas. But then as we make water unavailable again, then we condense the cells into these nematic droplets again. And so the nematic order correspondingly goes up again, all right? So I think that this is telling us that the capillary interactions are actually important for the colony to assemble these active nematic layers in the first place. And then that allows the colony to develop topological defects, which will then allow the colony to generate new layers and form fruiting bodies, all right? So this is what I wanted to say in this first part. So maybe let me just summarize by saying that I think the interesting aspect of this is that we found a piece of physics, which is capillary interactions, which together with activity allows this bacterial colony to aggregate, which is the first step towards forming fruiting bodies. And then I think that one interesting aspect is that through these capillary forces, you can form aggregates without cell-cell adhesion. And this enables the aggregates to remain fluid much more easily, and cells can move past each other because they're not attached to their neighbor. They're just together because of these surrounding capillary forces mediated by the meniscus. And then another point that I would like now to study is what's the role of these capillary forces once the cells want to form these three-dimensional structures, because then if you want to now move into 3D, you have to extend the water meniscus. And so at that point, capillary forces actually seem to hinder the formation of fruiting bodies, and that's an aspect that we want to analyze next, okay? All right, so are there any other questions about this part?
The sort of scenario and of going from through streams to fruiting bodies and all that is very similar to what is observed in certain fungi, at a larger scale than this, obviously. But is it, are the intercellular forces, etc., in Dictyostelium or something, are they similar to this, or is it totally different?
So it's very different. It's funny because for many years, the Myxo community thought the developmental process of Myxo was exactly like that in Dictyostelium. But they realized it isn't because there's no molecule mediating chemotaxis in Myxo. So Dicty is all about chemotaxis, and so they secrete a signal, they form these spiral waves, and that's a way for them to communicate over long distances and come together. But Myxo seemed to lack any long-range communication mechanism. So it's a much more physical approach to solving the same developmental problem. It's very different.
But it does have a shorter range chemical interaction.
It does.
Signaling,
Exactly, it does have signaling upon contact, but not long range. Yeah. So one here, yeah, and then there.
So how good is this approximation of quasi-static interfaces? I mean, if you model the thin film, you would get cubic mobility of the film, depending on the film type, the cubic weight and so on.
Yeah, I don't know. So we haven't seen.
Really slow research.
Exactly. So the cell motion is really slow. So in that sense, I think it's, you know, it does a relatively good job. But I'm not sure actually, because for very porous agars, actually, the dynamics of water could also be slow. And so in that case, maybe it plays a role. We haven't seen any effect that pushed us to study that in more detail, but I don't fully know. So that's a good point here. Yeah.
I was wondering about the formation of the holes in the first movie you showed. Have you, what's the mechanism for the formation of the holes, and have you characterized the statistics?
Yeah, very good. So actually that also has to do with the previous work that I didn't explain, but it also has to do with topological defects. So whereas +1/2 defects promote the formation of new layers, -1/2 defects promote the formation of holes. And so that's something we studied in the past, and we're also continuing to study to try to see how topological charge then gets delocalized along the periphery of the holes and so on. So that's how. Yeah. Yeah, Christina.
The reversal rate affected the holes by the extract of capillary force?
Not that we can see. We think that maybe friction is, because the capillary forces, in addition to mediating lateral attraction with other bacteria, they also push the bacteria down onto the surface. So we think that maybe this increases friction, or, you know, changes friction.
And then changes the reversal rate?
No, we don't think so. We don't think so. So the reversal rate seems to be controlled mostly by an internal clock, and this is mostly biochemical. And so this is what they might regulate either through the sensing of the availability of food or through contact signaling, but not mechanically as far as we've seen. Yeah. Okay, if there are no other questions about this part, then I'll move on to the second part. Feel free to ask about this again at the end. So in the second part, I want to talk about a completely different topic, and it's about flocking. And I want to show you a new mechanism of flocking that is based on turn-away interactions. So particles that turn away from one another manage to somehow move in the same direction and flock. So this is work that we did in collaboration again with experiments. So the experiments were done by Jie Zhang and Jing Yan respectively at the University of Science and Technology in China and in Yale. And we did the theory in my group at PKS together with a postdoc, Suchishmita Das, and with a PhD student actually of another group, Matteo Ciarchi. And so the motivation here is one that many of you are very familiar with, flocking. You see it in birds in nature. I hope that you've seen such a mesmerizing display as this one, in which many flocks in the sky align with one another and they fly together, mainly at sunset, as you can see. And so if you haven't seen it, here's a movie. Otherwise, try to go to Rome, where apparently it's the hot spot for these kinds of flocking events. And then the funny thing is that you can also see flocking in the lab. So you can engineer colloidal particles so that they become self-propelled, and then people have realized flocks with millions of these colloidal particles, as you can see here. This is in a channel in this case. And so what I want to discuss today is the mechanism of flocking. And so both in the case of birds and of colloidal particles, the mechanism that has been around since the work of Vicek and collaborators in 1995, so since the beginning of active matter, was based on saying that flocking is a result of alignment interactions, in which either the birds or the particles just align their directions of self-propulsion, and that produces polar order at the large scale. And so that's what I want to challenge today, and tell you about an alternative mechanism of flocking. And this we did by studying a particular experimental system, which is made of these Janus colloidal particles that have two faces. They are essentially glass beads, and you coat one half of them, one hemisphere, on a metal, in this case nickel. And so what you do with that is you take these particles, you suspend them in water, and put them in between two electrodes, and apply an electric field across them. And so then what happens is that the equators of these particles lie along the direction of the field, and so they all stand up like this. And then the electric field polarizes each of the hemispheres of the particle. But because one is a dielectric material and the other one is a metal, the polarization that you get in each of the hemispheres is different. And as a result of this difference, the ion clouds that you get around each of the hemisphere are also different. And this means that there's now an osmotic force that pushes on the particle and makes it self-propelled. And so particles like this self-propel, and that's a simple way of making self-propelled particles in the lab. And so what we studied first, and this is again past work, is what happens when the particles self-propel with the metal on the back. And so what happens in this case is actually my example of anti-diffusion. These particles start running when you turn on the electric field and they come together. And I won't discuss this because it's previous work, but this is essentially an example of motility-induced phase separation that happens through a completely different mechanism as compared to the one you're familiar with. It instead happens by torques that turn particles toward dense regions and make them aggregate into these dense regions. Okay? So this is past work. If you're interested in it, talk to me about it. It's the example of anti-diffusion that I wanted to discuss. And so what I want to discuss now is what happens if you now do... Yeah, questions?
Like Gauri Shankar and Rao years ago, where you have polarization which turns towards high density gradients and you have this kind of Astor lattices.
Exactly, but we don't have orientational order in the center of these clusters. So the center of these clusters gets a uniform density and therefore you lose the orientational order there. You only have interfacial order where you have the interface with density gradients. Yeah. All right, so then if you now rerun these experiments, but without salt in the solution, what happens is that you're changing the ion clouds around these particles and they propel in reverse. So now they propel with the metallic side in front, okay? And that's the case I want to discuss now. And so what happens in this case is at low concentrations, particles propel and they interact with one another, but they essentially form some isotropic gas. Instead, if you now do this experiment at high densities, what you see is something like this, a massive macroscopic flock of these particles. And as I was hinting at, this is actually very surprising. It looks like one of the flocks that has been realized in the past, but it's actually very different because if you think of the interactions between these particles, they shouldn't be able to produce a flock intuitively. And so the interactions between these particles are as follows. Imagine that we look at two of them from the top, and so you see the hemispheres from the top. The particles self-propel on the plane of the screen, and the electric field is perpendicular to the screen. It's pointing towards you. And so what this means is that the dipoles in each of the hemispheres are either also pointing towards you or into the screen. And so now if you work out the interactions between these particles, what you'll get is that the two particles repel each other electrostatically. And then because the dipoles are off-centered, they also exert a torque on one another. And so what happens is that the dipoles in the metallic side are stronger than the dipoles on the dielectric side, and so the heads of these particles repel more strongly than the tails of these particles. And this means that the particles turn away from one another. Okay, they want to place their metallic sides as far away as possible from each other. And so these are turn-away torques, and so the question is, how is it that they have particles that turn away from one another, and yet collectively they flock? Okay? And so to start to address this question, what we did was to write down the Langevin equations for this system. And so there's a Langevin equation for the positions of these particles, in which changes because of self-propulsion along the direction of self-propulsion n. Then there are these electrostatic repulsive forces and Brownian noise. And then there's also another equation for the direction of self-propulsion, which changes because of these turn-away torques and because of rotational diffusivity. And so then what we did was to simulate these equations, and these are simulations at low area fractions first. What you see is just an isotropic active gas, similar to what we saw in the experiments. And then at high area fractions, what you see is the development of these local polar order clusters or domains that then eventually coarsen into one big macroscopic flock again that spans the entire simulation, similar to the experiments. Okay? And so what we can do then in the simulations is to characterize this flocking transition and build a phase diagram. So here's a phase diagram in terms of the density of particles, the area fraction, and the self-propulsion speed. And you see that by increasing either one or the other, you go from the isotropic phase to the flocking phase. Okay? Now I want to take a small digression to tell you what happens if we go to higher densities. So in the experiments, we reach densities up to something like that, 0.25 area fraction. But in the simulations, we can go higher. And what I want to show you is that if you go higher, you actually get crystals at very high densities. Question, Silke?
Can you be a bit more specific exactly how your turn-away torque works?
Yes. So it's the torque is n cross r. So maybe I can write it. I'll show it later again, but essentially the torque between particle I and j is proportional to the orientation of particle I times the interparticle distance vector. And so one particle is trying to align its orientation away from the interparticle distance vector. And this is immediately non-reciprocal because if I switch I and j, then I'm replacing the orientation of particle I, but that of particle j, which has in principle nothing to do.
So is this completely not Physik style?
This is completely not Physik style. It's a completely different class of orientational interactions. They don't, you don't couple orientations, you couple orientation to position of the neighboring particle. Yeah. So what I want to show you now is just a few examples of, in the phase diagram before, I was showing what happens at low densities, and so the flux that you get are fluid, but as you increase the density as you expect, you then go through an exotic phase and then eventually a crystalline phase. And this crystal is a crystal that is assembled through the repulsive interactions between the particles and the fact that they live on a finite domain. And so this is what's known as a Wigner crystal, and it's, if you want, an active counterpart of the classical Wigner crystal due to repulsion between electrons in electron gases. Okay? And so I just want to show you a few snapshots of how the phases look like. So I'll show you these four. So the fluid flux looks something like this. What I'm plotting here is the orientation of the exotic order locally. And then if I go to the exotic phase, I have something like that. And then what's interesting is that once I go into this Wigner crystal phase, at low densities, I actually get polycrystalline samples. At low self-propulsion speeds, I get polycrystalline samples. But then as I increase activity, I mostly get almost monocrystalline samples. Okay? And so it seems like activity anneals the polycrystallinity of these samples.
When you say polycrystalline, what is the long time state there? I mean, if you
the long time state is monocrystal.
It is mono.
Yeah. But it gets stuck in these configurations, and it less so when you increase activity. Yeah. All right. So let me now go back to trying to understand the mechanism of flocking. How is that these particles flock? And so to address this question, what we did was to start from the Langevin equations and coarse grain them. We did that in two ways, using two types of kinetic theory. The first type was using the so-called BBGKY hierarchy that maybe you're familiar from liquid state theory. And so in this case, the final result of these calculations is to produce an equation for the polarity field of these particles with this kind of lowest order contribution, and in which we predict the value of this coefficient A. And if this is positive, then you can see that the polarity fluctuations will grow and the system will be destabilized from the isotropic state and generate flocking. If A is negative, then the isotropic state is stable. And so we predict this result for this coefficient A. It has two contributions. One that comes from torques, as I will show you now. This contribution from the torques is hidden in this coefficient tau naught. And then a negative contribution from noise that tends to hinder flocking. And so this tau naught coefficient is something like that, has this expression. You can see that it emerges from the torques, which is this functional here. This is the radial dependence of the torques between particles. And then there's this other function, which is the pair distribution function that tells me about correlations between two particles. And you can see it has three arguments. So let me define what they are. So let's imagine we have one reference particle here along a certain direction ni, and then a second particle here, which is moving along another direction. And so the first two arguments have to do with the distance vector. And so they're just the distance between two particles, r, and then the angle made by the distance vector and the self-propulsion direction of the first particle. Instead, the third argument here, theta, is just the difference between the self-propulsion directions between the two particles. Okay? So now, based on this, you can see that this is a bit of a challenge to measure in experiments because you have to sample a function of three arguments. So what we did was to actually measure it in simulations, which is also a challenge, but we managed. And so then we can use the simulation result here to obtain the value of tau naught at different area fractions and different self-propulsion speeds and predict when A switches sign from negative to positive and therefore predict the onset of the flocking transition. And so for Silke again, this is the expression that I now show explicitly of the torques. And so this is what comes here. And I'm showing it here just to show you that this cross product is responsible for one of these sign factors. And the other sign factor comes from a Fourier transform somewhere along the calculation.
Basically, is the mechanism that I have a particle to my right and a particle to my left, and I don't want to go towards my particle to my right and not to the particle towards my left, and therefore I point in front?
We thought so for a long time, and this is the argument that I will present in a second for the stability of the flocking phase. So once you flock, then yes, then you're a particle in the middle of the flock, you turn away from your right neighbor, you turn away from your left neighbor, and the compromise is to just keep going. Fully agree. But the question is, when you're in the isotropic phase, that's not the case. And so you have to find a mechanism to destabilize the isotropic phase and generate flocking in the first place. And so the argument there is different. So, all right, so that's what we did was just measure this function g of r, the pair distribution function in simulations, plug it in here, obtain tau naught, and then see when A changes sign, predict the flocking transition. And so this is how it looks like. This is what we had before. This is just the phase diagram that we built from simulations. And now we can superimpose the phase boundary that I told you about, and it lies here. So it does a decent job at capturing the flocking transition. And then what I wanted to point out is that once you flock, then exactly like Ananya was saying, the mechanism by which flocks are stable is just because if you're a particle in the middle of the flock, if you turn a bit right, you'll get closer to this particle, and then you'll turn away from this one, and so you'll get back to your original position and orientation. Okay? So it's a sort of a compromise between turning away from either side. What we did next was to redo the coarse graining calculation, but using a different type of kinetic theory that actually allowed us to get insight into what's the mechanism by which these particles flock. And so this we did by doing Boltzmann kinetic theory. And so in this case, what you do is you just consider pair scattering events, okay? Just binary scattering. And so let's consider the following. Let's imagine that these particles only interact via torques first as a simple scenario. And so let's consider a collision between these two particles, or a scattering event rather, between these two particles. They come into the scattering range when they go into the interaction range, and at that point they start turning away from one another. At some point they are parallel, but they keep turning, okay, and they'll keep turning and then moving apart from one another until they hit again the interaction range. And so because all of this process is perfectly symmetric, they move away from one another at exactly the opposite angles as they came in. And so there's no gain in momentum in a collision that involves only torques. So this cannot generate flocking at all. Now instead, if you now add the ingredient of repulsion that we also have, what happens is similar but crucially different. So when the particles come into the interaction range, they start turning away from one another, but also repelling. And so now they also become parallel, but they are further apart than before. And when they exit the interaction range, they haven't turned as much because they've been pushed apart, all right? And so now this collision process has generated some gain of momentum, and so these can now produce flocking. So you actually need not only turn-away torques, but also repulsion to get flocking. And that's quite surprising because in all the classical models of flocking, repulsion is not even in the picture. If you take the Vicek model, there's no repulsion, there are only alignment torques. And here I'm telling you that in our case, repulsion is actually necessary for flocking. And so to show this, what we did was to carry on the calculations with this Boltzmann kinetic theory and get the same equation as before. This is the equation for the polarity field, predict the same coefficient, and now it is given in terms of the same noise as before, but now here we have a quantity that comes from the collision statistics. And so this is the gain of momentum that I told you about in the previous slide, averaged over all possible collisions, which are parametrized by these two angles here. Okay, so what we do is we run simulations with just two particles in the world, so just binary scattering simulations, and average over all possible collisions, and get this coefficient here and see again when A switches from positive to negative. From negative to positive. And so we can do this by varying the repulsion between the two particles. So when they don't repel, we actually get a very negative result for this gain of momentum. So particles actually lose momentum through collisions on average. But when we start having more and more repulsion, this gain of the momentum is actually eventually positive, the momentum gain. And so particles start gaining momentum through collisions if they repel enough. And the mechanism is exactly what I told you before. And so, so this shows that repulsion is necessary, okay? And so now we can use these arguments to predict the phase boundary, and again, you know, it roughly falls in the same place as the other two boundaries, so this is all consistent. And so the final thing that I want to say is, what if you compare to experiments? So we had these experiments, we analyzed many of these experimental movies, and we just classified the ones that showed flocking versus the ones that didn't. And so the ones that showed flocking are marked with these filled symbols, and the ones that didn't are marked with these empty symbols. And roughly you see that our calculations match roughly what is seen in the experiments. Okay? So just two thoughts to end up. First is that when thinking about self-propelled particles, people often think of two big classes, which are either repulsion dominates the interactions, in which case the standard phenomenon that you get is motility-induced phase separation, or you think of alignment interactions, in which case you get flocking. What I'm showing you now is that we got flocking by some interactions that are not even alignment, instead turn-away interactions, and crucially, we needed repulsion, which was thought to be part of a different class of phenomena. Okay, and now we're kind of mixing and working in the boundary between these two classes. And so one final thought is that maybe, and only maybe, what I told you could be relevant for cells. And there's sort of a paradox in cell behavior, in that biologists describe the interactions between cells through a concept of contact inhibition of locomotion. So what they say is that when two cells collide, they collapse their protrusions, they retract this protrusion here, they form a new one, and then they move apart from one another. And so if you look at the process like this, it looks pretty much like the scattering events that we have in our particles. And so there's this paradox that says that even though cells seem to turn away from one another, they actually flock. And when you put many of them in a monolayer, very often you see flocking. And so whether what we did has to do with solving this paradox, I don't know, but I wanted to propose the idea and leave it open. And if you have ideas along these lines, please let me know. I'd be interested in hearing them. And so I just want to summarize again by saying that we found this new mechanism of flocking based on turn-away interactions plus repulsion, which has a key role. And I think that the kind of conclusion that we get is that flocking is really hard to avoid. Even if you have interactions that seem to kill it completely, they don't manage to. And so it's really a very widespread phenomenon. The relevance for cell colonies, I just wanted to pose as a question mark. And then I think one final message that I find interesting is that this is a very clear example of something that I believe is more general in active matter, which is that the symmetry of the macroscopic phases is emergent. We have here macroscopic phases with polar order, but we started with no microscopic interactions encoding that polar order. So really the polar order here came from nowhere, and it just emerged from the fact that these are active particles. And so that's how I wanted to finish. And I thank you very much for your attention, and I look forward to questions.
Questions so far?
One comment. So this turning away interaction, I think it maps to a known class of interaction which is called velocity or self-alignment, where you're mapping to the direction of the force or the velocity between them. And that means that has been used for cell colonies based on pretty much the same argument about contact inhibition, and it leads to blocking. I should also say, by the way, that would be really cool in an experiment. With that type of interaction, we put it in a confinement, it starts to oscillate.
Perfect, yeah.
For example, you see this Aggregation shows oscillating different material things. So this would be one way to use this further for back information.
Great, yeah, sounds good. I'm happy to discuss more of the details later on. Yeah, thanks.
I would also like to make a small comment and then ask you a question. The comment is that I really enjoyed the talk, and the story for this case where it's turning away and the repulsion is basically preventing it from turning away is like opposite to how we think about correlation due to self-propulsion interplaying with other things, because here the correlation due to self-propulsion is basically the correlation is preventing it from turning away from each other, like it's getting in the way rather than enhancing something that the system wants to do. So it's very pretty.
Yeah, in that
and
sense.
the question that I had is, what is the size of the simulations that you're doing? And the reason I'm asking is that my head suggests that you will get polar lanes of guys going this way and guys going that way, and it takes forever for them to find everybody going the same way state.
Yeah, so the simulations I showed you were the smallest ones with 2,500 particles, but I can show you bigger ones. We went up to 70,000.
Did you cheat in the smaller ones by helping it find the polar state?
No. Just, but let me show you the big ones because maybe they'll help clarify states that we get. So at low densities, for example, we get flocking without any help. The video is broken by the end, so you'll see strange things at the end, but it should go through. And you'll see, it takes a while, as you say, but eventually you see polar bands that start to emerge. And so these are the kinds of states that we...
So how many particles are there in this box?
In this box, 40,000. Yeah. And now is when the video is corrupted. But so we get polar bands which are very similar to the ones you get in the Viccheck model, but for completely different reasons.
Yes.
Was already asked.
Oh, okay. Good. Yeah, Jochen.
About the first part of your talk.
Yeah.
You showed this gradient from like wet conditions to dry conditions and how the bacteria would align differently. Bacteria, also cyanobacteria, they excrete substances. So they can influence the water balance, but they can also influence the surface tension. So basically, how they move is under their own control. Have you considered looking at that? It would, I think, be an interesting extension.
Yeah. So we thought exactly in this way, and we thought, it's great to have seen this because it means that now Myxo have another knob to play with to decide when to aggregate and so on. But we actually could not find any secretion of surfactants in the experiments, which was surprising. So either we just cannot measure it, or these bacteria don't do it, which seems silly because they lose a knob. But that's just what the experimental status of things is as of now.
Yeah, if you've got particles with a strongly preferred speed, and almost any interaction that makes them like to have the same velocity as their neighbors, you'll get a flock. An explicit aligning interaction is not necessary.
Exactly.
This is one manifestation of that in some sense.
Yes. That's maybe one extreme example because it's I would say quite surprising that even if they turn away, they essentially do what you say, and so even in that case you get flocking. Yes.
Right.
But to kind of go back to also what Silke said, I mean, this is precisely, I think, the interaction where the particles align with the force they feel due to their neighbors, which has been used actually in the context of cells first, quite a long time ago. Right?
Yes.
And it produces flocking and produces all the phenomenology of flocking.
Yes, but I agree on first pass, but I actually want to think about this better, maybe with you guys, because I'm not sure that everything matches.
You have to rename a few things, but it matches.
Yeah. Okay.
Well,
Great.
I think the question was from your gamma.
Exactly. Yeah. So overall, I agree, and then I'm happy to look at the details later on. Yeah.
Yes.
The question for the first part, for the experiments, you said that they are always killed if you add surfactants. But did they try also biosurfactants? I mean,
Yes, I cannot now recall exactly which ones, but yes. And somehow it didn't work. It was, we didn't expect it, so we thought it would work, but it didn't.
yeah, because there are other experiments like Bacterial subtilis where you can play around with surfactants.
Yes. I know, and that's why we wanted to try that first. It simply didn't work. I'm not sure exactly why, but the bacteria ended up dying in all cases. Maybe not because of the surfactant anyways, but yeah, it simply didn't work. And I'm happy to discuss exactly which one we should use to make it work and so on.
You probably need some kind of surfactant associated bacteria. There is a type of bacteria who love surfactants.
Okay, yeah.
Okay. Last one, perhaps.
So in the two-body scattering events, how did you get the effect of the density?
So what you do there is, let me remember.
You're tagging two particles, is it?
Yes, we simulate two particles, but the density is in the prefactors. So in this coefficient that I set, the density actually comes as a prefactor. So what we compute from the Boltzmann kinetic theory is just this average of the momentum gain, and then the dependence on the density comes from prefactors that maybe I didn't show. Yeah.
Okay. It's coffee time, so let's continue this discussion over the coffee. Thanks very much.